Hello, and welcome to each and every single one of you. As I'm so excited and so glad to have each of you today in class. Of course, I'm Mr. Harris from Grand Canyon University. In our class, of course, Chemistry 530, the concepts of biochemistry. Our topic today will be GPCR signaling, and I hope you have a somewhat of an idea as to what that actually is. If not, today we're going to break that barrier and today we're going to learn all about it. I'm under the superb direction of Dr. Karma Cook, and on this fine day, February 21st, 2024, I hope that you're ready to learn. And y'all, let's do it. Let's do it. Let's do it. Know that you have it in you. You can do it. So, of course, let's learn together and let's do this. Alrighty, class, get your pencils and pen ready and your paper, and we're about to start. However, before we start, we're going to do what we do every single class period, and we're going to discuss the lesson objective. So it states, students will be able to distinguish between the structure and function of GPCRS, differentiate between GPCR families, and identify pathophysiology slash the clinical significance associated with GPCRS. All right, so on your paper, what I would like for you to do, think about keywords within our objective. What words stick out to you? I'm going to pose an example. All right, so I see the word distinguish. I might know what it means, I might not. But that's a word that sticks out to me, so that's a word that I would write down. So any word that sticks out to you in our objective, write that on down. Furthermore, I have a very important question to ask you. The question states, what is the purpose of learning this objective? Why should I learn about the structure and function of GPCRS? Again, GPCRS is possibly a term that I've never even heard before, and that's possibly a term that I wrote down. I can somewhat say that may be an acronym for something. I don't know. I possibly have never heard of that one. So therefore, again, I want us to think and think and think. Of course, get in our mindset, of course, what we're going to learn. Get in the mindset, again, that I know that I can do this, even though it may seem difficult. So once again, on your paper, and I'm going to allow a few moments. So once again, figure out what words stick out to you. Again, what words are familiar, which words are not familiar. And we're going to come together and we're going to discuss those terms and we're going to figure out, and of course, it will come together as one. And we're going to discuss what's the purpose of learning about GPCRS, what's the purpose of their, learning about their families, the pathophysiologies, the significance, in a setting for clinics, and we're going to tie it all together before we really dive into our content material. All right, everybody, give yourselves a pat on the back, give yourselves a round of applause. Again, you all did a phenomenal, outstanding, amazing job. I'm so grateful for each and every single one of you. Again, I always say I'm definitely blessed to be here. And again, you all just prove more and more, of course, that you are more than capable and more than worthy of academic excellence. So to each and every single one of you, keep up the great work. I greatly appreciate how you all engaged in our turn to talk. I greatly appreciate how you all were engaged specifically in how, of course, this all came together. It was very interesting to hear your thoughts as to the words, again, that you selected. Again, I heard, once again, GPCRS. Again, I heard about some have never heard that term, some have heard it. And it's just interesting hearing the dynamics and the depth as to what some know and what some don't know. And it was very intriguing, again, to really understand and to really hone in on what you all stated regarding the purpose of learning about this objective. So once again, of course, kudos to you all. Again, today, I wish we had an observer today because today would have been a perfect day for that. So again, thumbs up to you all. And who knows, in the great book, I'll probably add some extra credit. So very good job to each and every single one of you. All right. So as we have our pencils ready and we learn and we know our objective and we know the purpose of it, today the breakdown as to what we're going to learn is as follows. Or are as follows, rather. All right, so it states the introduction. We have two slides of the introduction, and it's going to discuss cell signaling and what specifically are GPCRS. All right, and then we'll talk about the mechanism of signal transduction, the structure of GPCRS, the function of GPCRS, the families that are associated with GPCRS, and we're also going to discuss the pathophysiology and the significance simply of GPCRS. And at the end, we're gonna have a conclusion, and we're gonna discuss issues of concern again that arise with GPCRS. All right, so I'm so excited for today, and let's dive on in. Alrighty, class. All right, so we're gonna once again the first slides are gonna be introductory slides, and I do understand that for some of you all that this is going to be a review, and I know for some of you all this is going to be brand spanking new. All right, so all I do is ask of you again. I told y'all the only thing to pass my class is to try, and of course, simply, of course, to have a positive attitude. All right, so if you have those two things, you're already, basically already there. Again, know that you can do it. You've got it in you. I believe in you. I know that you can. And if you ever are feeling lost, again, let me know. Again, send me an email. See me see me after class rather. And we'll definitely work with that. All right. All right. All right. So I'm going to dive into our lesson for today as we dive into our introduction, excuse me. All right. So I'm going to first talk about cell signaling. As we see on the slide, again, we see cell signaling is an important process. Of course, that is required, that's mandated in order for cells to grow in a normal sense and to also grow in an effective and efficient manner as we, of course, continue, of course, to develop in our own lives. Well, we're also going to note right now that cell signaling it allows cells again to sustain in a multitude simply of environments. So to think about that as we ensue and as we go through. All right, cell signaling is just so important, and it is going to include and it also involves machinery again, which involves certain molecules. In other words, we're going to know receptor molecules and they're going to perceive signals such as light, possibly even, of course, an antigen, possibly a neurotransmitter, the service simply of another cell, and hormones, and also more simply in that aspect, in that respect. And also going to note again, very interestingly at this point, again, sometimes again there is sometimes what we know as a target, what have you. So the target simply of a signal again may include lots and lots of things, such as intracellular receptors, transcription factors, special transport vehicles, of the sort. And lots of times again they are controllers and they control gene expression. And furthermore, a vital signaling cascade. Again, that symbiote is going to be constructed, of course, by what we know as GTP, binding proteins. They may, of course, be classified simply as G proteins. And that's a part of our lesson and a part of our objective for today. And in other words, so the G proteins, as I just mentioned, sometimes they are given their name, and they are given their name specifically due to their ability and capability to connect again with what we know as guanine nucleotides. Also, furthermore, another major molecule, which of course today is our main topic, is going to be referred to as GPCR, or in other words, GPCRS, and of course, which can be referred to as the G protein coupled receptor. So once again, GPCRS, that stands for gene protein coupled receptor, and that's of course going to be possibly our main topic for today. Alrighty, everyone. All right, so once again, to emphasize and to reiterate, so GPCR or GPCRS, again, they're referred to as G protein coupled receptors. All right, and we're gonna dive into a bit more details about what those receptors entail. All right, so GPCRS, or in other words, G protein coupled receptors, again, they are the largest family that consists of human membrane proteins, and they have as many as 800 members. Furthermore, again, a GPCRS, they have the ability and the capability to detect an array of diverse molecules, and they also have the ability to trigger a multitude of cascades that deals with intracellular signaling. Furthermore, again, drugs are often involved, of course, with GPCR or GPCRS, and they may block, they may induce, they may even modulate a plethora of effects, as GPCRS are also known, once again, as large class of drug targeting. And we're also going to note, very important, and who knows, this is possibly a good test question. We're going to put a note that about or approximately 1/3 of drugs they're going to bind, they're going to connect to GPCRS, and they're also going to make specific modifications within as well. And furthermore, as again these receptors, again GPCR or GPCRS, and these receptors as they transmit signals again through the cell membrane, the ability of them to transform or to change its shape, again it must be considered simply as we ensue and as we go through. We're also going to note right now again that GPCR signaling is going to range from going to range from photons, ions, again proteins, and also much more. And once again, the human genome consists of approximately 800 again a GPCRS or G protein coupled receptors, and also and that accounts for approximately 3% of the entire genes simply of course the human species. And the GPCRS, the superfamily consists of subfamilies, again, which we will discuss momentarily. And furthermore, signaling such as, again, GPCRS protein expression, again, impaired ligand concentration, and others, again, they have been involved in a plethora of pathophysiological conditions, again, such as cancer, again, gastrointestinal disorders, cardiovascular disease, and more. And we're also going to note that GPCRS, they're integral proteins, and they also associate, again, with cytoplasmic side simply of the plasma membrane in that respect, in that regard. Alrighty, my ladies and gentlemen. All right, so now that we have some background information regarding GPCRS, the G protein the G coupled protein receptors, our job now is to figure out what are some of the mechanisms again that are associated of signal transduction, and also going to figure out how does that tie in, how does that correlate again with GPCRS respectively. Well, we see the figure right here on the screen. All right, so of course we got GPCRS. All right, and the figure right here, it displays the regulatory cycle that consists of G proteins. Furthermore, the subunits of the heterotrimetric G proteins, again, they associate with one another, with each other specifically. And they have states, and they can be inactive, and they can also be active, respectively. And in their inactive states, again, during a signal transduction, the GPCRS, it becomes activated. And also it becomes activated with a specific change. And the change of conformation, again, from the binding of ligands to certain regions simply of the actual GPCR. And furthermore, again, the G protein, of course, we're going to have what we call, certain things. And the G protein mediated signaling is going to begin by connecting, or in other words, binding the agonist molecules, again, which will eventually lead to, again, the GPCR in the state simply of activation. And furthermore, the GPCRS is going to promote, is going to trigger, is going to stimulate exchanges simply of guanine diphosphate and also guanine triphosphate as well. And furthermore, again, as this exchange is going to occur as it happens, again, the activated state simply of GPCRS is going to catalyze. It's going to catalyze the transfer simply of the GTP, or in other words, the GDP, or respectively in that way. And furthermore, simply if a G protein, of course, is activated in this manner, it's going to initiate an amplified, somewhat we could say somewhat an excited, possibly an intensified representation of the activated version of the GPCR, respectively. And also the activated proteins, they're going to bind, they're going to connect to multitudes of different effectors. And of course, we're going to dive in and we're going to talk about the GT pace. And the GT pace activity, it may be regulated by specific regulators of G protein signaling in addition to effectors. And furthermore, enzymes such as, adenoclases, again, they may affect and they may regulate the activation notion simply of G proteins by their own receptors. And furthermore, during the mechanism of signal transduction, the activated G alpha proteins, respectively, may connect or bind to effectors such as protein kinases, potassium channels, calcium ions, and so much more. All right, so everyone. All right, so our slide for this one is going to discuss the structure of GPCRS. All right, so GPCR is going to consist of superfamilies which share similar structures. They contain interstellar carboxyl terminal tails. Furthermore, these specific plasma membrane bound receptors, again, they have transformed, they have changed, and they recognize, again, a lot of chemical signals such as photons, calcium ions, peptides, nucleotides, and so much more. And furthermore, analyses of approximately 200 GPCR sequences have ensued, and the total length of GPCRS, again, they may range between approximately 300 and 1,500 amino acid residues. And furthermore, the large variations simply have been located in the C and N termini as well. And the sizes of those, that they range, of course, from approximately 879 to approximately 371 residues of amino acids. And furthermore, we're going to note that GPCRS, again, they may be encoded by viral and also eukaryotic genes, respectively, in that specific tone. And we're also going to note the alpha helices of GPCRS, they're connected, again, by 3 extracellular and 3 intracellular loops. And the extracellular loops, again, they may be glycosylated, and they may have 2 cysteine residues, again, which simply may assemble disulfide bonds to maintain the structure simply of the receptor. The N-terminus of a lot of GPCRS, again, are involved with ligand binding, with activation, and more. And furthermore, the helices of GPCRS, they're arranged, they're in tight ring-shaped core that are hydrophobic. In other words, again, water-hating. All right. And the hydrophobic amino acid residues are faced toward the core in lots of ways. And furthermore, the intercellular carboxyl terminal domains of GPCRS, they're involved in GPCR signaling. And already, so we've discussed the structure of GPCRS. So our job on this slide is to discuss the function of GPCRS. All right. And at this time, we're going to note, again, that GPCRS, again, they're referred to as surface receptors. And furthermore, again, they're involved in an array of physiological roles. GPCRS, again, they're involved in lots of things, again, such as immune system regulation, taste, smell, behavior, mood, and so much more. And furthermore, all aspects of GPCRS, again, they're going to involve specific extracellular signals that eventually convert or transfer into a cellular response. Furthermore, mood regulation, simply, and GPCRS, again, they are correlated. And they relate to each other in the sense that the GPCRS receptor is going to bring capability to bind or connect to a multitude of neurotransmitters. It may create lots and lots of physiological responses that may bind to GPCRS. And they may happen within the brain. It may lead to lots of different moods, again, such as serotonin or even dopamine. And furthermore, GPCRS is consistently in persistent atomic level motion, and even when the outside or external stimuli, of course, are not present. And already, as we are starting to wind down a tad bit, just going to go over some families of GPCRS. All right, we have 6 families, again, that are associated, again, with the G protein coupled receptors. All right, and once again, they are the largest class of receptors. And as it states on the first point of the slide, again, over 1,000 GPCRS have been identified. And that's just thus far. And so therefore, again, there are more that are consistently being discovered, of course, which is a good thing. All right, and once again, this is going to approximately account for 3 to 4% of human genes, and again, which is comprised simply of GPCRS. All right, we have 6 families, again, that are associated with GPCRS, and we're just going to briefly just discuss those right now. All right, so, and we'll start, of course, by letters, and we'll go from A, of course, through F. So family A, of course, is referred to as the rhodopsin receptor family. And out of all of 6 families, again, this is the largest family of GPCRS. And once again, it is going to contain small ligands and also going to contain odorants and also more. All right, family B, as we see, that's the secretin or the secretin or receptor family. It contains about 60 members, and it lacks structural signature, again, that family A possesses. And family B also has ligands of high molecular weight hormones such as calcitonin, certain growth hormones, got glucagon, and so much more. Our next family, family C, that's called the metabotropic uh, glutamate receptor family. And it consists of, once again, of these receptors in addition to calcium ion, you know, like calcium ion receptors, taste receptors, olfactory receptors, taste receptors, and so much more. Family D, called the fungus pheromone receptor family, and it contains again the pheromones as we see, and they also collaborate, of course, with GI as well. Family E, called the camp receptors, and they're only found in certain species, respectively. And also family F is called the frizzled smooth receptor family, and that's involved in embryonic development and also involved in cell polarity and also cell segmentation. And already to everyone. All right, so we're on our technically our second to last slide. All right, and as I spoke with some of you, some of y'all told me that you're interested in going to medical school. All right, so I know this will possibly be of interest to you as we discuss the pathophysiology and the clinical significance of GPCRS, or in other words, the G protein coupled receptors. All right, so we're going to note at this time that over 30 human diseases and syndromes, again, have been associated, of course, with the gain or loss of GPCRS. And they may cause a mutation. And so diseases such as autosomal dominant non-autoimmune hyperthyroidism, again, are caused by the activation of mutations, again, within, of course, the TSH receptor gene. We're also going to note at this time a gain of mutation LHR again has been identified, of course, to cause a gonadotropin releasing hormone in many GPCRS. And again, they may lose their function because of the friendship mutations, the nonsense mutations, again, insertions, deletions, and also more. And mutations that are involved in the rhodopsin gene commonly cause what we know as retinitis, and it may cause again retinal dystrophy in that respect. And also with that stated, again, so again, retinitis, again, pigmentosa, again, that's referred to as an eye disease. And with that one, of course, the retina of the eye becomes completely damaged. And also, people or individuals that have this, this mutation rather, they have difficulty with their sight in low light conditions. They have blurred vision, and at this current time, there is no treatment for that specific mutation for that condition. And also, again, hyperthyroidism, hypothyroidism, and other complications such as fertility issues and others, again, they may result, again, from mutations of GPCRS. And also, the G protein may be affected, of course, in not genetic. So, in other words, again, the G protein may be affected, however, and not be necessarily genetic. So, in other words, cholera. And we can think about the cholera outbreak that happened years and years and years ago. Well, cholera, it occurs as bacteria multiplies in the human intestine and secretes, of course, a toxin, of course, associated with cholera. And this toxin, it triggers cells that correlate with G protein, and that's one, and that's a takeaway again to dive in at this point. And all right, so ladies and gentlemen, thank you so much. All right, so as a conclusion, as we wrap up our lesson for today, all right, so just going to recap a few things. All right, throw in a note that GPCRS, they're integral, as we see on our slide. Again, they're integral proteins, and they comprise of approximately 3 to 4 simply of our of human genes. Furthermore, I've said this a billion times, and I was say it more. I'm gonna say it one more time. That GCPRS, the symbol, of course, means G protein coupled receptor. And of course, these receptors are classified as the largest group of membrane proteins, and also the largest of the drug targets as well. All right, so once again, GPCRS, again, they deal, of course, with ions, they deal with proteins, and so much more. And again, GPCRS, again, they may recognize photons, nucleotides, and more. And they're involved with regulation of the immune system, again, taste, smell, behavior, moods, and more. And furthermore, there are six families simply of GPCRS, and once again, there are in approximately 1,000 GPCRS they have been discovered. The loss or gain of GPCRS, again, they may lead to mutations, and furthermore, issues of concern in future studies. Again, they indicate simply that every structure has not been found of GPCRS, and furthermore, more studies are definitely needed in order to fully understand and comprehend the structure of GPCRS. Again, only 1,000 approximately have been discovered, and of course, there are more to be discovered. And in current times, again, the only understanding comes from computational and also experimental research additionally. So therefore, those are issues of concern that possibly will be addressed in the future. So definitely take that to heed. And here are references. I thank you all so much for an amazing class. Again, thank you all so very much. If you have a question, feel free to email me. Feel free to see me after class, and I will see y'all next time. Y'all have a wonderful day. All right. All right. Thank you.